This conference will now be recorded.
Okay, folks. So, uh, technical issues. So we're looking at the blood chapter. So today we're going to be discussing and looking at one aspect of the cardiovascular system. The cardiovascular system in lab there, we've been looking at blood typing. So we did blood typing, we did blood pressure, as well as cardiac anatomy and such, and review of the heart anatomy as well as the blood vessels. And so, and also the vascular flat model that we looked at and the muscular head. Okay, so you all, let's see, so you will have, I don't have access to my, let's see here. On, just to confirm that there, yes, so we will not have, so next week on, that would be the 15th, we're going to have group B, lab group B in the lab. They will have their last lab before the practical exam. On the 22nd, we will not be in school because there'll be our spring break. And then the 29th, both groups, lab group A and lab group B, will be on campus, and we will take turns with test number 2. So you'll switch. One will be taking test number 2, while the other group will be taking the lab practical, and then you'll switch, and one will be taking the practical as the other's taking the test number 2. Okay, so very good with that. So, and as can't hear me. Okay. Michelle, there must be something. Let's stop. I'll stop what I'm doing. So Michelle can't hear me. Can everybody else hear me? So give me some feedback first, folks, just so that you can hear me, Norma? Okay. So
I can hear you.
All right.
Lisa can hear you.
Very good. Thank you, folks. So Michelle, it's got to be something on your end. You might want to exit out of the class and then come back into the class. Know that I'm recording, so you won't miss anything. Okay, so try that again. All right, so thank you folks for your input and helping us out there. So let's go back to the PowerPoint. So we're looking at the blood. And so the importance of the blood, we all understand the importance of the blood, because realizing that if there's some type of traumatic injury and we lose the blood that is present within us and we lose enough of it, you can go into shock and die as a result. Know that we need this blood circulating through our body and oxygenating the tissues, in particular the brain, right? The neurological tissues, the central nervous system, the brain and the spinal cord. Without that oxygen, tissues will die. Without, as a result of a cardiac infarction there, a myocardial infarction, heart attack, the distal area where the blocked off blood vessel is not reaching, right? Blood's not reaching, that tissue will die. So we know the importance of having oxygenated blood and just blood in particular, traveling throughout the whole body and reaching the systemic circulation, all the parts of the body with this oxygenated blood, and bringing also nutrients and moving many other materials other than just oxygen and food and glucose. So we're going to look at that today. So let's first take a look at this first slide here. No, yeah, so understand that. So you have your quiz. So that's just the quiz as far as the circulation through the heart. That quiz will be on this Monday. Right, remember, so let me just make this point again. So the week when we come back from our spring break, both groups, lab group A and lab group B, will be on campus. And I think, right, I said that was the 29th. Sorry, folks, let's just take a look at here again. Yeah, the 29th, Monday the 29th, lab group A and lab group B will be on campus. So you'll both be taking the test. You'll alternate between taking test number 2 and taking the practical examination. And I'll go over that, Jen, at the end of class, okay? Yeah, very good. So I will make mention of that again at the end of class. But know that it's going, yeah, I'm not going to deal with that right at the moment. So, all right, so as we look at the blood, no problem. All right, so as we look at the blood, know that a good portion of the blood is fluid, okay, 55% approximate, right? And then the formed elements, when we think of the cells that comprise, that are present within and flowing within the blood, we think of the red blood cells, the white blood cells, and the platelets. The proper terms would be the erythrocytes, right? Leukocytes and the thrombocytes. know that these are these we call the formed elements. And again, the plasma, that's the fluid portion of the blood. And know that the fluid portion of the blood is extremely important in that we're making sure that as we're having circulation and interacting with the capillaries throughout the body, that there's going to be plasma leaking out of the capillaries and into the spaces of the body in order to allow for the lymphatic system to do its role, as far as that plasma will be quote unquote sucked up, brought into the lymphatic vessels and become lymph, okay? And that will be a part of that lymphatic system that's involved in the ability to really act as a surveillance, looking for pathogenic molecules and pathogens that could make us sick, okay? And so they'll be passing through, flowing through lymph nodes and flowing through the lymphatic organs and such, and then coming back to the cardiovascular system via the right atrium, okay, and returning to the blood. So the fluid, so a portion of the fluid can leave, right? Can leave the cardiovascular system, but then it returns to the right side of the heart and into the right atrium. Know also that as far as blood is filtered in the kidneys, and when that filtration takes place, a filtrate is formed, which will become urine. Now, quite a bit of blood is filtered throughout the day, but only a small portion of it actually exits the body, excreted from the body as urine. Primarily, the majority of it, 99%, is reabsorbed into the body, okay? But the blood is vital for life. You'll see here, as far as we're looking at the percentages, as far as the plasma, again, approximately 55%, and then the formed elements, approximately 45%. And out of that 45%, the majority of those cells are going to be the erythrocytes. Only a very small portion, folks, will be the neutrophils, the eosinophils, basophils, the lymphocytes, the monocytes, the thrombocytes. So the white blood cells and the platelets, right, the thrombocytes, only a very small percentage, like 1% or so, if that. Really, it's the erythrocytes that comprise the majority of the formed elements. So as we mentioned this, and I talked about this right from the very beginning of the semester, you need to know, as far as when we go over each chapter, what the function of the system that we're looking at, and in particular, we're focusing on blood, one aspect of the cardiovascular system. So what's taking place with blood as far as its functions are concerned, right? So involved in transportation and transportation of oxygen primarily, right? This is the key issue, oxygen and nutrients, and the removal of CO2, waste gases and waste products, and transport of hormones, right? These chemical messengers that are transported throughout the body, produced in glands, and then sent into the blood, and then delivered to specific receptors for those specific hormones throughout the body. Regulation of body pH, regulation of body temperature. When we have vasodilation and we have the blood as coming to the surface, very close to the surface of the skin of the integument, we're going to release a lot of heat if the temperature is high, okay? And if the temperature is low, then what's going to happen? We're going to have vasoconstriction, and really we're going to keep most of the blood going to the vital organs and to the brain, and not so much to the periphery. So that's why it also, when you're outside in the cold, your extremities are going to be cold because your body's reacting to the cold temperatures as such to protect and to provide proper circulation to your core, to all the organs present within the core of your body, as well as your brain. The clotting mechanism, dealing with thrombocytes, these platelets here, and as far as the different chemicals that are involved in that whole process and such, as well as these tiny little pieces of cells, okay? Protection, and so know that your cardiovascular system works in conjunction with the lymphatic system to provide immunity for your body. So the immune system, when we talk about the different body systems, the immune system is a functional type system. It's not an actual system per se in that, right? So like your cardiovascular system, your lymphatic system, those are absolute systems of the body, and they work in conjunction with each other in order to provide the immune system of your body, okay, and protect you from illness and such, and combat any type of pathogens that your body may come in contact with. The maintenance of body temperature, working with the thermostat of your body, which is the hypothalamus. And osmosis, and so really the movement of water. And I said to you that the importance of the plasma in the blood and how that is very important for, one aspect I didn't mention was, so I talked about, I discussed it as far as the lymph, this fluid that's present in the lymphatic system and present in the lymphatic vessels. I talked about the formation of urine. I didn't talk about the production of cerebrospinal fluid, and how also that from the blood and from the liquid portion of the blood, working with specialized cells present within the glial cells present within the central nervous system, we're able to produce cerebrospinal fluid. Very important. And maintaining this osmotic balance, this balance of where the water moisture is present throughout the body. Know that things can get out of kilter and homeostatically be out of balance when a patient has congestive heart failure and there are issues with edema, swelling within the extremities. Okay, so we'll talk more about that in when we look at the heart, and also when we're looking at the lymphatic system. So you see here, as far as the red blood cells, white blood cells, the platelets, erythrocytes, leukocytes, and thrombocytes. 95% of the volume of the blood cells, you know, and greater would be the, and it's an approximate, right, are the red blood cells. The leukocytes, the thrombocytes, very important. Thrombocytes, again, these are involved in for your blood clotting mechanism and such, and for sure, Uders. So know that cerebrospinal fluid, right, in conjunction working with ependymal cells. Remember this in the central nervous system. The ependymal cells will interact with blood vessels present within the choroid plexus. Do you recall that? So the choroid plexus present within the ventricles of the brain will interact with the ependymal cells and create CSF, cerebrospinal fluid. Okay, and so that's as a result of the liquid portion of the blood becoming a part of that, becoming that CSF, that cerebrospinal fluid. Okay. Now you'll see here, as far as there are granular and agranular types of white blood cells. You're welcome, Urus. So we have here, as far as the granular ones, are the NEB, neutrophils, eosinophils, basophils. The agranulocytes, right? So agranular, agranulocyte, also known as monocytes and lymphocytes. Okay, and we'll discuss these in further detail. Now, as far as the numbers that are present within the blood, so we have the majority of the white blood cells are the neutrophils, the highest in number. The lowest in number would be the basophils, right? So we have this little silly mnemonic that says never let monkeys eat bananas. And so what will occur is that we have the majority of our white blood cells are neutrophils. Then we have the next highest number would be lymphocytes, monocytes, eosinophils, and lastly, the basophils. Okay, so that can help you just as far as knowing, hey, what is so when you're actually in the lab and we'll be looking at blood smears, you'll find that you won't see so many basophils when you're looking as far as scanning and looking at the slide, but you will see many neutrophils. But even then, when you're looking at the blood smear, primarily it's all red blood cells. And then, but just as far as when you're scanning and you're seeing those purple stained cells, primarily they will be neutrophils as far as the white blood cells. And you'll see little tiny specks of cells, and those are the platelets. So here you're looking at and you're seeing an example of, and know that the monocytes, they are the biggest cells. They are the largest cells as far as the blood cells present. Here you have the lymphocytes. Now know that when we're talking about the immune system, that the lymphocytes, there's going to be B and T lymphocytes. And we'll discuss more about them as far as, in particular, where those white blood cells mature, as far as whether it's in the bone marrow or in the thymus gland, and hence the terms B and T lymphocytes. But these lymphocytes, vital role in your specific immune response. Can you remember that? You should be writing down notes when you're in my class in the lecture here, and I'm saying a lot of different things. And when you review if you do review the video write notes, folks, and you can pause it and stop it and go back. But I do give you more information than just what's on the PowerPoint here and such, okay? So you'll see here as far as that, again, like I said, the monocytes, they're the largest in size. And you'll see very unique presentations of these cells, right? As far as when you're looking at eosinophils, they have a very unique presentation, and we'll look at slides in the lab and you'll see how you can differentiate, and you will have to differentiate in the lab. As far as for practical number 2, I will be asking you information regarding, no, practical, wait a second. So we're doing the, that's right. I did not go over with you all in lab there as far as the blood smear as far as for lab group A. I can't believe I forgot to do that. So we were doing a lot of other things as far as taking the quiz and as far as going through some different things. So what I will do, folks, is I will create a video, right, regarding, so I'll do that tomorrow. I'll create a video regarding the histology of blood. Okay, so of a blood smear, and I'll create this video, and so I'll share it with you both classes. And then what I will do is I will open up the lab. Now you won't come into the lab, understand that, but you can come into the room number 214. If that, now I know that this is not an easy thing, so like I'm just giving you that opportunity because I forgot to. Yeah, Kayla, I'll go over the, ask me at the end of class, please. And I'll definitely do that. I'll go over that with you all. So I will offer the opportunity for you all for lab group A, to come on campus and be in room 214 in order to view slides, the blood smear in particular, okay? So I'll give you that opportunity to do that. All right, so yeah, so I do apologize that I didn't cover that. So that will be also, so the blood identification of the blood will be also as far as in the lab practical number 1, because it's not lab practical number 2, it's lab practical number 1. And you will have, in addition to the endocrine histology, you'll have I'll put up on the smartboard images of blood, and you'll have to I'll point out, hey, what is this, identify this blood cell. Okay, and you'll have to be able to identify whether it's a lymphocyte, monocyte, eosinophil, neutrophil, basophil, red blood cell, platelet. I won't ask you red blood cell and platelets because they're very easy to identify. But I will ask you a couple of questions, maybe like 2 questions or so regarding the blood cells. Okay, now what you're seeing here as far as the monocyte is concerned, see how it has this horseshoe or kidney-shaped nucleus? That's the monocyte. Very, very easy to spot and to see as far as you'll see. I'll show you images later on. And again, too, in that video that I'll record, I'll share that with you all. The lymphocyte really is primarily the majority of the cytoplasm is taken up with this oval-shaped nucleus. Okay. Now, when we get to a neutrophil, the neutrophils, these are the granular sites, right, or the granular types of leukocytes. You'll see that it has multi-lobes, like more than 2, okay, more than 2 lobes, and it's very unique in its presentation and pretty easy to spot and to look at, okay. The eosinophil and the basophil, I'm going to just have to show you images of so that you'll be able to identify and see the difference between them, okay. Now, the fluid portion of the blood, again, high percentage of water. There are going to be plasma proteins and some solutes present, okay? So the hormones can be a part of those solutes. Enzymes, which are proteins, they can be traveling in the blood. Waste products, gases, right? So know that oxygen, CO2 carrying through there. Nutrients, ions, all part of that plasma solutes. The plasma proteins, albumin, globulin, and fibrinogen. Fibrinogen for clotting, globulin for one aspect of those globulins are the immunoglobulins, which are your antibodies. Okay, and then albumin. Albumin, it's the produced in the highest amount as far as the plasma proteins are concerned, and it allows to have really that you have these solute concentration present within the blood, in order to allow for and to prevent really the majority of the plasma, the liquid portion of your blood, leaving the blood vessels and going out into the tissues of the body. So having these solutes present, these proteins as well as these other solutes, like I just mentioned before, will help to retain the water and not lose all the water to osmosis to the outer tissues of the body. Hematopoiesis or hemopoiesis. This is the production of blood cells in the red bone marrow. Okay. Know that infants and children have a higher percentage of red bone marrow present within the body. Primarily, adults will have it in the flat bones of the body as well as the epiphyses of the bones. Okay, the ends of bones, and also like the pelvic bones, they would be the areas where red bone marrow would be present. All other areas would be the yellow bone marrow, which is adipose tissue storage, but red bone marrow will produce blood cells. Okay. Stem cells are the undifferentiated mesenchymal cells that are present, and you'll see as far as there is, let's see if we have that image here on this one. Okay, yeah, I didn't provide it because I wasn't going to really. All right, so there are different stem cells that will produce the different blood cells present within the blood, simple as that, and they will become only those specific types of blood cells. Erythropoiesis would be then the production of, in particular, red blood cells, erythrocytes. Now, erythrocytes are not long-lived. You know, approximately 120 days. So you're looking at about approximately 4 months that they'll last. And you know, that's again, that's an approximate, right? But that's their duration. And then what will happen is that they will be recycled. Okay. Now, know that red blood cells that are circulating and doing their job as far as transporting oxygen to the tissues of the body, they are anucleate, meaning that they do not have a nucleus. Okay, so here's what I was going to mention to you regarding their specific stem cells that will then produce, and this is the different cells that are produced until we get to really this reticulocyte, which is an immature red blood cell. A reticulocyte is an immature red blood cell that will become an erythrocyte. Okay. The hormone erythropoietin, okay, the hormone erythropoietin stimulates red blood cell production. Okay, so when the body, through homeostatic mechanisms, realizes that we are low in red blood cells, then the body will release more erythropoietin into the system in order to stimulate erythrocyte synthesis, okay, and erythropoiesis. Now, can athletes, would they be involved in trying to do what they can to stimulate this as far as wanting an increased level of erythrocytes flowing through their body? Well, yes, they could. And there's blood doping. There are different techniques that are done in order to actually take, either by methods of injecting EPO or really giving transfusions of their own blood prior to an event, some type of athletic event, to increase the amount of erythrocytes flowing through their body, excuse me, in order to provide better oxygenation of the tissues of the body for that specific athletic event. This is dangerous. This is very dangerous, and this can cause a heart attack, a stroke. There are many risks involved with this, but it's been done. In particular, Lance Armstrong was one of the ones that was involved in doing this in hotel rooms prior to sporting events and such, and so to races. Pretty crazy stuff. Yeah, because you know, we know that most hotel rooms are also can be used for operating rooms, and I mean, seriously, that's crazy stuff. Not very sanitary. Yikes. All right, so erythrocytes. Yeah, that is insane, isn't it? Seriously. Well, you know, I mean, I understand competition. I get that. I totally get it. Very competitive person myself, but realizing that, you know, there are rules and there are safety issues also that we need to follow. Excuse me, let me get a drink here. All right, so erythrocytes, right? So these mature red blood cells flowing through the body, right? Bioconcave discs, anucleate, no nucleus. Contain 1/3 hemoglobin, 2/3 lipids, ATP, carbonic anhydrase. Involved in the primary role is transportation of oxygen to the tissues of the body. There's a small percentage that will be involved in carrying CO2, waste gas. Oh, I'm sorry, I've been talking a lot over the past few days. Carrying CO2 to also, right, transport of CO2, but really what will happen is that CO2 primarily is being transported in the plasma and not so much on the actual erythrocytes themselves. Okay, you'll see here as far as what goes on as far as the heme and globin molecules present in hemoglobin, this protein, and iron being present in order to bind to oxygen and as well as CO2. So it's a pigment, right? Hemoglobin is a pigment. It's a protein molecule made up of globin, an iron compound called heme, right? And so iron is very important. So if we have an iron deficiency anemia, can we have issues with what's going on as far as carrying of oxygen to the tissues? Absolutely. And we'll go over, I have like a 3-page document that I've posted for you all. It's already been in there as far as we'll talk about blood pathologies, okay? So oxyhemoglobin, carboxyhemoglobin, this is when we're carrying oxygen, when we're carrying carbon dioxide, this is what's going to be taking place as far as the hemoglobin and carrying of those specific gases, okay? So oxygen, 98.5% is attached to hemoglobin, only 1.5% in the plasma. So a very small portion of the oxygen is being transported in the plasma. You'll see here, so now I said to you that a good portion is being transported in the plasma, right, the CO2. What I didn't say was, so let's hear me out for a moment here. So 23% attached to hemoglobin, right? That's in this, so carried by the red blood cell. 7% in the plasma, and then 70% as bicarbonate ions also present within the plasma, okay? So you're looking at really 77% that's carried in the plasma of CO2, okay? Only 23% carried as far as attached to the hemoglobin, carboxyhemoglobin, okay? So neutrophils, eosinophils, basophils. You should know, and you're going to need to know as far as what it is that each of them function-wise are they present for, okay? And I will, excuse me one moment here. Yeah, because I go over it in lab. Yeah, okay, all right. So I'm going to step away for a moment from the PowerPoint. I'm going to just give you a brief introduction regarding the white blood cells. And like I said, I will dedicate tomorrow. I'll take some time and create a video just for identification of the specific types of blood cells present, okay? So here's something that we can take a look at. Let's see if this is a little bit better. That's a good one, okay. All right, very good. So let's save this image. I can, okay, folks, so this is good here. So take a look at this for a moment here. And so what we're looking at, folks, is that here we have, right? So here's an eosinophil, here's a neutrophil, here's a basophil. So would you want to, like, this is going to be in the video. So write this down for me, please. So A, neutrophil. B, eosinophil. C, basophil. Right? So these are the agranulocytes, right? I mean, these are the granulocytes. I apologize. These are the granulocytes. Now, these are the agranulocytes. Here would be a lymphocyte, and also what are you seeing here? Right here, right here, right there, platelets, okay? So lymphocyte and platelets, thrombocytes, and then we have here a monocyte. See how much it's larger, and also a very unique pattern as far as the nucleus is concerned, okay? So this would be a monocyte. Now, know that after a few days, monocytes will leave the blood and become macrophages. So they differentiate. So monocytes will become macrophages, and macrophages are involved in phagocytosis as well as neutrophils, okay, involved in phagocytosis. But the macrophages, they are really one of the key, what's called an antigen presenting cell, an APC. And as well as the dendritic cells and macrophages are involved in becoming an antigen presenting cell, which will really be a very crucial, important part of the immune system. Just give you that for now, but know that very important aspect of the immune function, okay? And know that the lymphocytes, B and T lymphocytes, are actively involved in your specific immune response. B and T lymphocytes are involved in your specific immune response, okay? So again, as we're looking at these images, need I point out to you what these are? These are your erythrocytes, your red blood cells, okay? So neutrophils, eosinophils, and you'll see here the neutrophil has multiple lobes to its nucleus. You'll see here that the eosinophil, right? So when you think of eosin, so you think of the color red. And so look at this cytoplasm of the eosinophil, it's red. Now you look at the basophil and it's really hard to differentiate between the nucleus and the cytoplasm, okay? But now don't confuse that with the lymphocyte, because you can see that there are kind of multiple lobes and it's a little bit more granular, hence these are the granulocytes. And here we have the agranulocytes. And so the lymphocyte really, it has like a crescent moon of a cytoplasm. The majority of the cell is comprised of this oval-shaped nucleus for the lymphocyte. And again, like I said to you before, the monocyte, the largest of the blood cells, of the formed elements, and it's kind of like a horseshoe-shaped or a kidney-shaped nucleus. Okay. Excellent. So when I create this short video for you all regarding the blood cells and looking at different samples of blood cells, and that's what I'll do. While I'm creating the video, I'll show you multiple images of blood smears, and I'll identify the different blood cells to you. That'll be helpful. Okay. Let's minimize this. Let's go back to our PowerPoint. So now coming here, so let me just stop for a moment. Okay. So I'll just ask, are we doing okay? Is everybody all right? Do you have any questions at all at this point in time? Or I know we'll address, we'll talk about test, we'll talk about quiz, we'll talk about the practical. I'll go over things, review everything with you at the end of class, okay? But any other questions at this point in time?
Yes, sir, please, can you kindly please repeat about the 7% plasma for the carbon dioxide and like the 77 that you were repeating, please?
Yeah, so it's just a matter of, okay, so let me hide everyone. Yeah, so it's just a matter of just realizing that there's a process that's taking place that will actually, so there's a process that takes place within the blood where we will have an interaction between the erythrocyte and CO2, and part of the 7% approximate of the CO2 will interact with the red blood cell, and it'll interact with the carboxyhemoglobin, right? And it'll stay attached to the red blood cell. The other 70% approximate or so will interact with the red blood cell and create bicarbonate ions. And then when the red blood cells that contain the bicarbonate in the plasma and the 23% attached to the hemoglobin, and also the 7% and then this would be what? Deoxygenated blood returning back to the right atrium. When it hits the lungs, right? Hits both of the lungs, and in the capillaries surrounding the alveoli, this is where then the bicarbonate will come out of solution, and CO2 will come out of the bicarbonate, convert with the red blood cell, and become CO2 and be exhaled out into the lungs, into the little air sacs, these alveoli that are present within the lungs. So it's just know that what we want you to know at this point in time is that 23% attached to the red blood cell itself, directly attached to it. This 70%, this is going to interact with the red blood cell and then become bicarbonate. So CO2 will interact with the red blood cell, become bicarbonate through a couple of chemical reactions, right? That'll take place. And so then we're going to, once that bicarbonate and this deoxygenated blood hits the capillaries of the lungs, it will come out, CO2 will come out of solution, will not be bicarbonate, will become CO2 and be exhaled into the lungs, into the alveoli of the lungs. Okay, you're welcome. So as far as the, let's go back here, neutrophils are concerned. So think of the, really, I've heard it termed bacterial slayers, and that's a good one, just because I'm like this fantasy guy, and so I'm thinking like the neutrophils are like these warriors, right, that are out there. And I'm going to share with you when we come to the immune system, there's a very cool animation. I'll share a link to you, and I'll ask you to just write a little summary of it. But I think you'll find it very interesting as far as showing you in some animation as far as the immune system and how these different cells do their jobs. So it's phagocytic, meaning that it's involved in endocytosis, right? So this endocytosis material will enter into the cell. So phagocytizes foreign substances. Bacteria is the big deal here as far as those little microorganisms, right? That can, not all, but some can make us very sick. Some are pathogenic. The antibody complexes. So what goes on is that antibodies will attach to particulate matter that can make you sick. We'll leave it at that right now. And know that then the neutrophils can identify these and phagocytize them, okay? Lysozyme secretions, they're present within, okay? So lysozyme would be present in your tears, in your saliva, and again, you know, they're not lasting very long, to be honest, 1 to 2 days, because they can be really cause destruction. So we don't want them to be so active, the neutrophils, that they're attacking our own immune system, our own body, right? To become autoimmune, creating a chronic inflammatory state. It's not good. So neutrophils are very potent, very important as far as the immune response, but really more in part of the non-specific defense, okay? Eosinophils, they'll contribute to the allergic response. So and also, well, I'm sharing it with you all right now. I'll stop sharing for a moment, and let's see here, okay, and then I'll share it with you again. All right, just give me some feedback, folks, before we move on. Can everybody see my PowerPoint? Just give it. Okay, very good, Norma. Thank you, Kayla.
Yeah,
Thank you, Nicole. Yep, so everybody is seeing it, so that's got to be on your end. So you might want to check your Wi-Fi connection, but know that, again, I'm recording this, so anything that you've missed, you'll be able to review and see, okay? So I'm sorry, though, but yeah, sometimes we can have technical difficulties at home, and again, if there's issues with our Wi-Fi, that can be a problem. So, so you'll see here as far as neutrophils, very important. Eosinophils, again, so I think of parasites, and I don't know, like how I think of it is that a parasite at the end has an E for it there. So I think parasite, eosinophil. I don't know, that's a silly thing that I came up with as far as helping you understand and remember that they're associated with eosinophils. And know that if they're producing antihistamines, eosinophils, then they're contributing to the allergic response that you would experience, okay? As far as, or diminish, sorry, I apologize. The basophils are contributing to it because they're sending out histamine. The eosinophils are producing these antihistamines, so they're being produced in greater numbers, the eosinophils, as a result of the allergic response, okay? So the basophils are contributing to the allergic response. So you know this, right? You have an allergic reaction, what do you take? You take an antihistamine, do you not? Because this is going to help to diminish the allergic response. So that's what eosinophils, if there's an allergic response taking place, you're going to have more eosinophils present in order to produce antihistamines in order to calm down that allergic response, right? Whereas in basophils, they are actually produced, right? And again, basophils are the lowest in amount as far as numbers are concerned, and they're actually present during the allergic response in inflammation, allergic reactions. They're contributing to it by sending out histamine, as well as the heparin there will contribute to the flow of blood during this period of time and such. And we'll talk more about heparin and such in a little bit later on in the presentation here. Monocytes and lymphocytes, I mentioned to you regarding lymphocytes, how they are involved in the specific immune response. You might want to write that down next to it there. The T lymphocytes have we say that they're involved in the cellular immunity, right? And the B lymphocytes involved in antibody production. So the cellular response will be, in particular, cytotoxic T cells, helper T cells. These helper T cells are involved in really the specific immune response and really enabling the specific immune response to take place and really activating it, okay? And we'll talk about this again when we discuss the immune system. And also there are inhibitory T lymphocytes that will be involved in, once the infection has been taken care of, to then really suppress the immune response, the specific immune response. And for both T and B lymphocytes, they have a long life because there are memory B and memory T cells that have a memory to their encounter with specific antigens, okay? And so they can live for many years, all right? As far as the monocytes are concerned, I said to you that over a few day period of time, the macrophages will exit from the blood and go to the tissues of the body, go to specific organs present within the lymphatic system, and they'll be present there in order to wait for any pathogenic type of organism, infected cell, in order to phagocytize them, okay? So phagocytosis, bacteria, cellular debris. So the monocytes and the macrophages are like the cleanup crew of the body. Really, it's quite important what they do. And really, by phagocytizing and taking care of cellular debris, they're also involved in really recycling what takes place as far as what's present within the body. This term, Diapedesis, Chemotaxis, Amoeboid Movement, these are just movements that are present as a result of the white blood cells doing their thing, okay? Diapedesis, the cells will thin and elongate and actually move between or through the capillaries and into the tissues of the body, okay? Chemotaxis is really what we would describe as far as this movement of the cells to an area of inflammation, of damage, and these chemicals will draw, really, it's like a breadcrumb trail, so to speak. These chemicals will be released from the damaged site, from the site of infection, and it will draw the cells to that area, okay? And amoeboid movement would be the use of the pseudopods and stuff. So when we think of like macrophages and such and monocytes moving, that's kind of like that method of movement. Inflammation. So when we think of inflammation, know that inflammation is not a bad thing. Truly, your immune system, if it's functioning properly, you will have and you will begin an immune response. Will be one of the first immune responses will be an inflammatory response to some type of damage, right? Some type of infection. This is important. It's just when the inflammatory process is extended for a period of time, this can be a problem, okay? And so chronic inflammation is not a fun or good thing. And it can really lead to misery as far as the joints are concerned, as well as the cardiovascular system and other parts of the body where inflammatory process is not healthy for extended periods of time. So redness, heat, right? So the sensation of heat, swelling, edema, pain, and also, and we can add a fifth one here would be lack of function or a diminished function wherever this is taking place. So if there's inflammation in my joint, Will this diminish the function? Absolutely. So you're going to see it looks red. It's going to feel hot to the touch. It's going to probably be swollen. It'll be inflamed, right? There's going to be edema present. And as a result of the chemicals being released, as well as the swelling of the tissues, the edema present, there's going to be pain and discomfort there. And again, lack of function, okay? That's the inflammatory response. Learn that now, and that'll be important as far as when we're also talking about the immune system, okay? So these platelets, these thrombocytes, they're cellular fragments with a nucleus, which is quite interesting, right? So even though they look like they're just like little particulate matter and tiny cells, they have a nucleus present, and they are from a megakaryocyte. So this megakaryocyte, it's big, it's a big cell that has pieces of it come off with nuclei that are these thrombocytes, these platelets, okay? And as far as the clotting mechanism is concerned, so I'm not going to ask you to memorize all this, but I'm just going to show you as far as what's taking place, and know that within this process here, so we have chemicals that are released in order to pull and draw different other cellular debris, so blood cells, so platelets, red blood cells to the area of damage. And we actually have also, when you think of, we say fluid, that blood is a fluid connective tissue, right? And we know that connective tissues, we realize that they have different types of, like, let's see here, as far as elastic fibers, as far as collagen fibers, right? You could actually see them when you're looking at connective tissue. Well, in the blood, these fibers will only come out of solution when there is some type of damage. Okay, so these fibers will be present and will come to the area of damage and contribute to the whole clotting mechanism. Okay, so the clot, this fibrin, forms long threads acting like a net in which the platelets and red blood cells will come to and get enmeshed in and create this clot that will lead to the stoppage of bleeding from the damaged blood vessel. Okay. Now, over time, and as that the area is now, so the clot is immediate. That's very quickly, right, taking place. But over time, as the blood vessel is healing, so then we'll have a retraction of this clot, and that's called syneresis, okay? And then the actual dissolution of the blood clot would be fibrinolysis, these terms, okay? Now, these terms, and they're also present on my Word document that I'll be reviewing with you in a few minutes there. The term thrombus is a clot. The term thrombus is a clot. Unwanted clotting taking place is the thrombosis. And we'll see as far as embolus and an embolism, we'll see that the embolus is an actual clot, a thrombus, that's circulating. And an embolism is where we have this, this embolus stops in a vessel and it blocks the flow of blood. That's a very bad thing. So the term infarction or myocardial infarction for the heart is concerned, but an infarction just period, tissues are killed as a result of loss of blood supply. So wherever the embolus, the embolism is stuck in a blood vessel, whatever's distal in that blood vessel is no longer then receiving the blood because it's blocked, and as a result, tissue death will occur. Here you're seeing as far as the clotting mechanism is concerned. So you see here as far as the fibers present, as far as the platelets and red blood cells that are creating that clot, right? And so it's a matter of different chemicals coming together, drawing these cells together and creating this clot in order to stop the bleed of this damaged vessel. And know that also we talked about that as far as negative and positive feedback. This would be a positive feedback mechanism that would be healthy within the body, because we're continually sending out more chemicals and receiving more cells to the area until the point where we have this damaged area filled with the fibers as well as the cells. So now we did in lab blood typing. Okay, and so we're going to talk a little bit about blood groups and such now. And so know that there are different types, and there are different types of antigens present within the blood, but we're talking about the ABO grouping, okay? And as well as the Rh positive or Rh factor, and whether it's a negative or positive, we'll discuss that also, okay? Now the term agglutination, I mentioned this to you, those in lab on Monday there, regarding the clumping that's taking place. And what happens is that if you receive a blood type that is not your correct blood type as far as a transfusion is concerned, this can be a problem, folks, and we can end up having agglutination or clumping of your red blood cells. And if this clumping takes place within your blood vessels, can this also lead to an infarction, lead to a blockage of blood flow in the smaller tiny vessels? Absolutely, and this can lead to death. As well as, think about this, as well as you're not, then if the blood vessels, if the blood cells are clumping, are they going to actually be transferring oxygen to the tissues and removing the carbon dioxide and such and be involved in that process? No. So this is a very bad so this transfusion reaction can lead to death, okay, if it's not handled and taken care of, right? So we have so this don't look at this here, that's not a negative. So A, B, and O, and then we can have Rh positive or negative. So I'm A positive, so that's my my blood typing, right? And there are antigens and antibodies that we're going to discuss, as well as we talked about the agglutination, the Rh factor we'll discuss, and know that, and you need to know this. Oh, okay, very good, excellent. So universal donor, Rh negative. Universal recipient, AB positive, okay? So the AB positive, they can receive all the blood types as far as transfusion. Yeah. Understood. Yeah, Jen. Yeah, thank you for sharing that. Yeah, so that's a very real thing. And so we're going to discuss that as far as the issues with when this can affect a mother and child, okay? And in particular, the child, because the mother will could mount an immune response to her own child present within, right? So if mom is Rh negative and baby is Rh positive, this can be an issue. And so that ROGAM and such is very important. Okay? So let's look at the blood types here. So type A blood contains, and I didn't write this here, so let's, has, no, I don't, oh, you're going to say that's negative. There are, let's see, comma, A antigens are present. I'm going to make that smaller. Type B blood, there are B antigens, and I'll talk about this in just a moment. Type AB blood has A and B antigens are present on the plasma membrane of the RBC. Okay, let's make that even smaller so this way it'll stay in the same line. There we go. That's good. Okay. Now, AB antigens. So PM is the plasma membrane, right? Okay. Okay, so let's take a look at this again. So what's going on, folks, is that type A blood is type A because it has these A antigens present on the plasma membrane of the red blood cell. And present within the plasma, and let me write that down also, antibodies. I'm only going to put it for the first one. Anti-B antibodies present in the plasma of type A blood, okay? Okay, so let's come back here. So type A blood has A antigens present on the plasma membrane of the red blood cell, and in the plasma would be these anti-B antibodies present, okay? So when you think of type A blood, it's going to have A antigens and B antibodies. When you think of type B blood, type B is B because it has B antigens on the plasma membrane of the red blood cell, and it has A antibodies present, right? So this would make sense then if I have type A blood and I receive type B as far as a transfusion is concerned, what's going to happen? Well, my B antibodies present within my plasma will attack those type B blood cells, erythrocytes, and this will cause that clumping to take place, folks. This is why agglutination takes place. Now, if I have type A blood, AB blood, can I receive type A and type B? Yes. Why would I do? Why? Because I have A and B antigens present. There's no antibodies for A or B. So type AB, that makes sense that it's the universal recipient, okay? So this is not going to cause a reaction. So if I receive type A, if I receive type B, and I am type AB blood, it's okay. I don't have any antibodies for A or B, and I have A and B antigens are present on my erythrocytes. Now, type O blood, right? No antigens present on the plasma membrane. Now, notice that like there are other antigens that are present on red blood cells, but these are the most important ones regarding transfusion and such. And so that's why we're talking about these, okay? But there are A and B antibodies, okay? But know that really this is not an issue that we can have type O negative be the universal donor, okay? So let's take a look here as far as Rh, and then we'll look at an image that I think will be, yeah, I don't know, I didn't have it posted here. All right, so I'll show you an image. Yeah, in just a moment on the internet there. I'll look that up there for you. So Rh antigen, this is also an antigen, right? As a result of the rhesus monkey and testing that it was done and such, that's how we developed this. Okay, the R antigen, whether it's present, it's positive. If it's not present, it's negative, okay? So antigen G, okay? So anti-Rh antibodies develop after exposure. So mom, who is Rh negative, right? And she's carrying an Rh positive child. Here's the deal. If she is Rh negative, that means that she does not have the Rh factor. The baby does because father, the dad, had, within his blood and within the genetics and such, was able to pass over type B, positive Rh factor to the baby, right? And so what happens is that mom has what? So let's take a look here for a moment there. Mom has the antibodies present because she doesn't have the antibodies present at first, but once she delivers, that first pregnancy, she delivers, there's going to be an interaction between her blood and the baby's blood, not just the placenta wise, but through the cord, but actually through the birthing process, there can be an exposure to the baby's Rh positive blood. And what will happen is that mom, after that first exposure, then will develop antibodies because she doesn't have the Rh factor. So she'll develop antibodies for that, and subsequent pregnancies, mom can develop an immune response to an Rh positive baby. All right, this is called Erythroblastosis Fetalis, and this is a problem. So RhoGAM, very important. So if there's a determination that mom is negative, the baby is positive, and if there's any you know, if the dad, as far as blood typing is concerned, has a positive blood type, then but they're testing for this, folks, and they're figuring out, okay, if there's an issue or not. And did you have it? Wow, Jen. And you had the RhoGAM, and you and babies were well. So I'm glad for that. That's good stuff. So how cool is this, right? So years ago, prior to this, yeah, truly. Yeah, thank you for sharing that too, Jen. Understand that this was a major problem, and it's still in other, so we're a first world country, but in third world countries, is this an issue? This can be an issue if there's no type of exposure to any type of prenatal care, and so we can have these issues take place, and you know, not the first pregnancy, but subsequent pregnancies with an Rh negative mom and an Rh positive child can lead to you know, death of the child, because mom is mounting an immune response to the baby. So let's take a look here as far as what's available online regarding just some basics as far as blood typing. And I just want to show you as far as the antigens are concerned, right? So this is just a very simple image regarding, you know, just, yeah. So here, what you're just seeing, folks, just to show you, is that for this, as far as, and I can post this for you all, okay? I'll post this for you, this image. But you'll see here, so this person is a type A blood, type B blood, type AB, and type O, okay? So what we're seeing is that type A has these A antigens present on the plasma membrane of the erythrocyte. Type B has these B antigens present on the plasma membrane, and it will have these A antibodies or these B antibodies, right? Now, type AB would make no sense to have A or B antibodies because both antigens are present. You can see both antigens on the plasma membrane. Now, how about type O? A or B antigens are not present, right? And so A and B antibodies are present in the plasma. So I'll post that for you all on documents and resources, okay? And folks, you have to give me a moment. I have to step away from the PowerPoint presentation just for a moment. Give me just like a 3-minute time period. I'll return in just a moment. I'm just going to shut off my All right, folks, very good. So as far as the blood testing is concerned and such, so I'm not going to ask of you and it's not highlighted as such as far as giving me the actual what's going on as far as the different types of tests that can be done and such, and what's taking place as far as numbers and such, because I don't feel that at this point in time that's really going to help you all. And you're going to be learning that as far as in your respective training, as far as going into the nursing programs and such, or whatever other types of allied health classes that you'll be involved in as far as for careers. So I don't just go through that all. Okay. The disorders, I'm going to now stop the PowerPoint and then share with you all as far as this document that I provided for you, this Word document. It's called Blood Disorders. And you'll see here as far as, so I have for you all listing here as far as erythrocyte disorders, as far as white blood cell leukocyte disorders, platelet disorders, and here we have, oh, look at this here. So thrombus, embolus, embolism. We talked about this earlier there, right? So please review that, go over that. You'll see here as far as anticoagulants are concerned. So we are going to have patients coming into the office that are going to be on different types of quote unquote blood thinners, okay? And prevention really as far as aiding people who have cardiovascular disease and are prone to strokes and heart attack, right? And so these are given to those patients in order to prevent them from, to lower the risk of having a heart attack and stroke, okay? And also post treatment of heart attack and stroke to prevent further and allow for easier flow through the cardiovascular system as far as allowing for the blood to move easier, okay? Getting back to when we were talking about earlier there as far as the Lance Armstrong. And so understand that the viscosity, the quote unquote thickness of the blood, right? Imagine if there are more erythrocytes, red blood cells present in the blood. That's going to make it move slower, right? Can this also, in someone who's unhealthy, right, this could absolutely lead to a heart attack or stroke. But even those that are in very good physical condition, it also poses that risk, and it's very dangerous, folks. So really important to not play home doctor. And they'll even use people who have, you know, special training and such, but still it's cheating, and it's not right, and you shouldn't be doing it, and it's dangerous. So that blood doping is not a wise or smart thing to do. Here I mentioned to you, it apparently, Jen, it does. Apparently it does. It really does make a difference. And, you know, if you'll notice that, and really like, and know that, you know, so the Americans got caught doing this, but know that they, not that it makes it right, but that they all do this, right? That they, and there's been studies show that all the competitive teams do these types of things, and it's not right. But you know, people, there's always going to be cheaters out there, folks, and it's not a good thing. It's not a way to be. So you'll see here as far as hemophiliacs are concerned and hemophilia, so A and type B, as far as missing different types of clotting factors that will really inhibit the ability for them to clot as a result of any type of bruising. So they can have major bruising take place as a result of minor injuries that you and I wouldn't be a big deal. But so folks that suffer with hemophilia, it's not an easy life. And know that really when the blood supply was not as protected as it is now, many patients came down with HIV as a result of the blood supply in the early stages back in the 80s, the early 80s, not being fully safe, and and having people who were donating that had HIV present within their blood, and it wasn't being screened out. And so that was a pretty sad thing, that folks that weren't even involved in risky behavior, just having an illness, ended up getting HIV. And it's very sad. Yeah. So hemophilia, A, clotting factor 8 is missing, B, clotting factor 9. Okay. And know that there are multiple clotting factors. I think there's up to like 13
It shows you're on mute, Dr. Pearl. You need to unmute yourself.
All right, can you hear me now? I do apologize. This is the first time I've ever used my phone for this app. I don't know why my phone just my computer just stopped. So I'm going back into rebooting my new computer. Very frustrating. I'm sorry. And I will continue on with the lecture in just a moment. Okay. So just bear with me for a couple minutes, folks. Okay. Thank you. So my phone is my computer is booting up. I do apologize. Okay, let me see. I think I'm on here. All right, so. Okay, let me turn this off. All right, can you folks hear me? Are we okay here?
Yes. I can hear you.
I do apologize. I'm sorry. My computer all of a sudden just went, that's it. And it said there's a problem and we're rebooting. I'm like, what? So I'm sorry, folks. I do apologize for that. All right. So yeah, right. Yikes, it's true, honestly. All right, so let's see here as far as, let me share my screen with you all. Actually, where's that? Excuse me, one moment. I'm sorry. All these windows up. Okay, where is it there? So you'll see here, folks, I'm sharing my screen with you all. You can see that. So we have here the blood pathologies. So this is the one. Many of you have already downloaded this. Here we go. Okay, so you'll see here. So let's I'll just record a few minutes of this. It's not going to be too long, but I want to finish up the presentation. So I'll share with you the link to the heart as far as the video that I uploaded, okay, to YouTube, as for the anatomy for the heart, as well as this the video that we just had that I was abruptly removed from, and then this short video regarding the end of the class for today. So thank you for bearing with me.
This conference will now be recorded.
Okay, so we kind of adapt and go from there. You'll see here, as far as this document is concerned, so this Word doc for the blood disorders, really very helpful. I hope it's helpful to you all, that it gives you a lot of information in only a couple of pages as far as the Word doc is concerned. I'll just go back to the end for a moment here, and we had talked about regarding hemophilia, the coagulants, clotting going on, and now let's go back to as far as the erythrocytes, the red blood cell disorders. So you're all familiar with the term anemia, right? So this can mean that the blood has an abnormally low oxygen carrying capacity. there can be different causes for this, the etiology, whether there's an actual blood loss. So could there be a traumatic blood loss as a result of some type of injury, or say, go ahead, Norma, question. Go ahead.
Oh, my mute button was on. For hemophilia, you said there was up to a certain amount of them, not just like one or two before
No, so
everything froze?
those two types of hemophilia that I want you to know about, but there are
Okay.
multiple types of, let me move this here. There are multiple types of clotting factors. So you're seeing here
Okay.
8 and 9, but there are like 13 clotting factors. They're not like that many hemophilia types of diseases, disorders, but I'm just telling you about the two that I want you to know about, and that I'm just giving you additional information regarding that there are multiple types of clotting factors present. It's just those two correspond with the hemophilia A and B.
Okay, thank you.
You're welcome. You're welcome. So as we look at the anemia again and look at the different etiologies, so this low oxygen carrying capacity of the blood can be as a result of, like I said, traumatic blood loss. So, you're involved in an injury and there's blood leaking out of your body as a result of, major cuts throughout your body or loss of an appendage, right? Or as a result of, say you have an ulcer, some type of internal bleeding that you're unaware of, and that you're slowly losing blood over time, and you're not even realizing that there can be anemia as a result of that, or low red blood cell production or high red blood cell destruction, right? So it's affecting the red blood cells in some manner, okay? That you're having this ability to not carry the proper amount of oxygen to the tissues of the body. This can lead to fatigue, to paleness of the integument of the skin of the face in particular, shortness of breath, chills. Why is this going on? Because you're not getting enough oxygen to the tissues of the body and such, okay? So that hemorrhagic anemia, chronic hemorrhagic anemia, so hemorrhoids and ulcer can be the cause of this. As far as low red blood cell production, so iron deficiency anemia, I mentioned that before because iron is necessary in conjunction with the hemoglobin in order to carry oxygen to the tissues of the body, and in order to carry the oxygen on the oxyhemoglobin in the red blood cells. Pernicious anemia, this is kind of an odd one as far as an autoimmune disorder, destroys the stomach mucosa. Okay, well then how does that affect what's going on with red blood cells? Well, the lack of intrinsic factor needed to absorb B12, which in turn, as a result of B12 not being present at the proper amounts within the body, can lead to red blood cells that don't divide, and so that can lead to lower levels of red blood cells, mature red blood cells circulating in the blood. Aplastic anemia, this can be as a result of drugs, different types of chemotherapy, chemicals, radiation, viruses can lead to this aplastic anemia, which can lead to destruction of the red marrow and inhibition of its ability to produce blood cells in particular these red blood cells. Okay, let's move on. So then destruction-wise, so a hemolytic anemia. So when we think of lysis, we think of destruction of the plasma membrane of a cell and the death of the cell, okay? And premature red blood cell lysis as a result of some type of process going on as far as issues with the hemoglobin, issues with an incompatible transfusion. So, I have type A blood and I receive type B transfusion, that's a problem, and infections. This is also something interesting. These next two here, as far as Cooley's anemia or thalassemia, this is, do you really? See, in my family also, I have a few members of, not my personal family, but my extended family. We have thalassemia present. And it's a typical Mediterranean ancestry as who has it, but not necessarily. But there's issues with, as far as the red blood cells here being thin, delicate, deficient in hemoglobin, and this can, the severity can be from mild to severe, just like any other type of disorder that we can suffer from. Mild, moderate, severe. That's how we describe as far as what's taking place with the disease process and what the patient is experiencing. Sickle cell anemia, this is when we have the hemoglobin present within the red blood cells. When it's exposed to low levels of oxygen, when there's low levels of oxygen, they will become crescent shaped, okay? And the way to help this would be to have increased oxygen input, which will help to prevent this crescent shape from the red blood cells from becoming this crescent shaped. Understand that these crescent shaped, these sickle cell red blood cells can get stuck in, can clog the small vessels in the joints of the body and can be extremely painful, folks. And this can be, if not properly managed, this can be a really catastrophic illness. This term, polycythemia vera, again, we're looking at red blood cell destruction, and so bone marrow cancer, folks. This is we're actually. Oh, I'm sorry, Jasmine. That's terrible, and that's horrible. Yeah, I've seen a video of patients going through an exacerbation or a crisis with sickle cell anemia, and oh my gosh, my heart goes out to them. It's just so much discomfort and pain, and it's just a horrible, horrible illness. You'll see here, as far as bone marrow cancer can increase production of red blood cells, severely increased blood viscosity, right? I talked about the whole blood doping thing and how that's not a very positive or good thing, okay? White blood cell disorders, as far as leukopenia. So leukopenia would be the opposite of something like a polycythemia, in that there's excessive blood cell production, in particular red blood cells. Leukopenia would be low levels of white blood cells present, and this could be drug-induced as a result of some type of chemotherapy. Now, know that leukemias, this would be analogous with the polycythemia vera, in that we have an abnormal amount of white blood cells flowing through the cardiovascular system in the blood, and these can have, we can have acute and chronic types of leukemia, and these can, some can be very slow acting, and some can be very fast acting, and some affect younger children, and some affect older population. Know that it doesn't matter which leukemia that the patient has, over time, if untreated, it will be fatal, okay? And let's see here. And then lastly, as far as these two disorders here, as far as platelet disorders, as far as thrombocytopenia. So thrombocytopenia, there's a blood cell, the platelet deficiency. So the deficiency, there's not enough platelets. So can this also be an issue as far as clotting is concerned? Absolutely, right? So bleeding, bruising, slow blood clot formation. Petechiae appears. Now, let's show you this as far as spontaneous widespread hemorrhage. Let's take a look at this image here that I've shown. So look at these images here as far as these little micro bleeds, right, in the body. And these can be pretty pronounced and such, and this can be, and so if this is viewed on a patient, this is of concern. And I have a friend who is an older patient who he has he has thrombocytopenia, and he does have this on his body. And when issues are, when it's really bad, when there's issues where it's untreated and such, he can have a presentation like this, and he knows that he has to go to the hospital in order to get transfusion and such. Okay, so let's stop sharing. All right, folks, so I'm going to bring everybody out, and then what I'm going to do, folks, is I'm going to I'll continue this this actual recording so that I can go over with you all as far as testing purposes are concerned, and I'll pull up the syllabus in just a moment. But what I can tell you is that this coming up Monday, Group B will be involved in doing you're just going to have a blank piece of paper, and you're going to have to write out the blood flow through the heart. So from the superior vena cava, inferior vena cava, bringing deoxygenated blood to the right atrium, and then the valves and the chambers and the vessels that will be passing through, going through pulmonic circulation and then to systemic circulation, and also include coronary circulation. So we have from the aorta to the what? The right and left coronary arteries. Then from the coronary arteries, we go then to cardiac, and this is how basic I went, to cardiac veins, right? So the cardiac veins. Yes, yeah, you should, yes, absolutely, yeah. You're gonna have to know pulmonary arteries are taking deoxygenated blood, right, to the lungs. Pulmonary veins are bringing deoxygenated blood back to the left atrium, right? You need to know that. And let me go back to coronary circulation. So it was coronary, so the aorta, the first branch of the aorta is the right and left coronary arteries. Then you go to cardiac veins, the great cardiac vein, the small cardiac vein, the middle cardiac vein, draining deoxygenated blood to the coronary sinus. The coronary sinus will drain that deoxygenated blood from the heart through the ostium, that little tiny hole in the right atrium, to the right atrium to allow for it to enter into that whole circulation. Okay, so that's what you need to give to me. So however you graph it out, or if you just go one step at a time from the top of the page to the bottom, that's fine. You know, it doesn't have to be any crazy way. I just want to make sure that and confirm that you know the flow of blood through the body. Okay. Any questions with that right now? All right, then let me hide everyone. I'm going to share my screen. I'm going to pull up the, let's see here. Let's see. I'm just going to go to the website and just again download. I'll just get the syllabus and then we'll just go from there as far as looking at. Okay, so let's move this up here for a moment. Okay, so what's not on here as far as for lab practical number 1, right? So lab practical number 1 and group A, again, like I said, you can come in and be in room 214, and I'll have a couple microscopes in there for you to look at blood smears and be able to identify the different types of white blood cells in particular. Again, tomorrow I will create a short video that I'll again post the link for you all regarding identification of the different types of white blood cells and just show you more images than I did during the PowerPoint presentation, the lecture presentation today. But know that, right, the sagittal head, neurological components, the small brain, right, will be on it, as well as the eye and the ear, the endocrine system histology, the heart, the flat model that will contain the blood vessels, right? You'll have to identify. The muscular head has some blood vessels, and please, lab group B, ask questions when we're in the lab on Monday, please. And then also, no, not yet. I'm sorry. Yeah. So because I do have the final, actually 2 out of the 3. So I still have one more person that's left that needs to take the test. And so I don't want to release the tests. And really, it wouldn't be fair because then only part of the class would have the test before the yeah. So sorry about that. So I'll make sure that you both get it, you know, soon. Okay. But because we have spring break, that kind of messes the flow of things a little bit. And know that as far as on the practical number 1 also, so I'll just ask like 2 questions regarding blood pressure, taking the blood pressure, and also regarding blood typing. So I'll have 2 questions regarding blood type on the practical exam. As far as I'll have 2 little plastic trays that have a specific type of blood that I've set out here that I've done the lab for, so that you can see, all right, is that A positive, is that B positive, is that AB positive, AB negative, O negative, O positive. You'll have to determine that by looking at the 2 little plastic, sorry folks, my phone's going off there. You'll have to be able to identify that regarding on those little, and you'll see that lab group B, you'll see that coming up this Monday, okay? All right, so that's it for that. Then as far as the test is concerned, so again, test number 2, let's see here. So test number 2, yeah, I'm not going to tell you that right off the bat. There was just knowing as far as how to actually properly take a blood pressure. So I'll ask a simple question regarding locate what artery would be important as far as to be able to listen to when you're taking blood pressure. Understood? So like the brachial artery. That's simple as that. Just nothing ridiculous regarding blood pressure taking. Okay. Yeah. Okay. So as far as test number 2, it's going to be on the autonomic system, right? So the third PowerPoint, the endocrine system, and blood. Okay, so that's going to be test number 2. I should have had test number 1 to include the autonomic nervous system, and I usually do. I didn't do that this semester, but I'm going to do that next semester because it creates a little bit of an issue as we get into the semester. So it is what it is for this semester. So you had first test was on just the first 2 PowerPoints. The next test number 2, will be on the autonomic nervous system, endocrine, and the blood. Okay, and then the next test will be on. So we're going to have 4 tests total, and know that those tests would drop the lowest score. Okay, so any questions now? So let me stop sharing for a moment here and just see if there are any questions that you all have. I'll bring everybody out. Okay, so again, that test will be after the spring break. Okay, so you have plenty of time. Please, I would tell you, so let me take some moments here and discuss with you regarding what you should be doing as far as your studying is concerned, right? Because if you did poorly on that test, the first test, and I don't want you to do poorly on any of the subsequent tests or the practical exams, you need to do something different about your study habits, okay? And primarily, I'm going to tell you one of the big things that you're not doing is you're not studying enough, okay? 2, you're not managing your time as well as you possibly can in order to, and I'm not, this is not judgmental. This is just a way that I want to share with you how I think that you can do better, okay? You also need to be effective as far as your time management is concerned. And when you are studying, that you are spending the time really where you're not having any distractions, maybe even, so I have these that I use a lot, right? These are little, let's see here, earplugs. They're foam and they go in both your ears there, and really you can zone out everything that's going on around you and just focus on what it is that you need to study. So very important, folks. I have to tell you that that's really crucial and important to have your time period where you're at your computer, at your desk, 25 to 30 minutes at a shot, and you're just focused. Then after 25, 30 minutes, take a break, go do something else for 5 minutes, come back. 25, 30 minutes, focus. Then again, take a 5-minute break. So you'll do this for a few times. And then after like 3 times of doing that, now this is like on a Sunday or Saturday when you have extra time, then you can like take a half-hour break and then come back and start the process again. Sitting at the computer for an hour or 2 at a time and not moving, that's never productive. You need to give your brain a break for a few minutes and then come back to it again. If you're not taking your PowerPoints and rewriting them, you know, that's a big deal. I'm going to tell you that right now. That's really helpful in order to rewrite the information that you're seeing. There's something about copying work that really helps you to memorize the information. And so like you can take notes in specific types of terms and rewrite them over and over again. Let me stop what I'm doing here. So what I'm sharing with you, how many hours a day do you suggest we study? So Nimrose, that's really, it's up to you all. I don't know, and I can't tell you. Some people can do, you know, certain periods of time, and some people can't because of their life and what's going on. But know this, you know, that really effective studying every day will help you as far as get into the program that you want to get into. All these programs are competitive, folks, right? No matter what it is that we're trying to get into, whatever school we're trying to get into, and primarily it's nursing, it's competitive. And so you need to do as well as you possibly can in these courses in order to get into the program that you would like to get into, right? So, you know, yeah, so Ruth says here, I listened to these in the car on the way to work. Yeah, so really passive learning by listening to lectures, by listening to videos regarding the structure, the anatomical structures in the lab, listening to my videos, other people's videos regarding the subject matter that you're studying. Yeah, and I highly recommend you listen to other people also, because just hearing someone come at a subject matter from a different point of view can be very helpful. But know that, you know, I know you all are making sacrifices to be here, to study, and you want to do well for yourselves and your families. I come here, I honestly, I give as much as I can, 100%, to help you all, but you know, I can't do it for you. You have to figure out what's going to work for you, and also you can pursue tutoring. You can pursue different websites online that can be helpful, but the fact of the matter is you have to do something about focused study every day, you know, and whatever that's going to work for you, I don't know. You have to figure that out, but I'm going to tell you, I would recommend like an you know, hour, an hour and a half a day that you're studying. And if you're doing it 7 days a week, every once in a while you miss a day, but on some days then you do more. But you really, you have to study every day in order to process the amount of information that we ask of you, okay? I just have to tell you that that's a big deal as far as doing flashcards that are actually that you make of the terms that you don't know. So you're at a disadvantage in that many of you are learning medical terminology while you're taking this class. And you know, with A&P1 and now A&P2, it's hard, folks. It's not easy. but again, right? You can do it. You just have to put the time in, and it does require a lot of sacrifice. I know that. So if you would like to talk to me more about that, I can help you as best as possible. Okay. All right. So I'm going to stop recording right now.